Welcome back, everybody. Good afternoon. I'm Dr. Bobby. Welcome back to our daily lectures on New Age Cosmetic White EdTech Healthcare Consultancy. We are on the subject of molecular biotechnology, and in particular, we're on the topic of directed mutagenesis and protein engineering. On to the subject of DNA shuffling. Well, biologically important proteins such as alpha interferon, IFN alpha, as in chapter 10, are encoded by a family of several related genes, with each protein having slightly different biological activity. If all or at least several of the genes or cDNAs for a particular protein have been isolated, it is possible to recombine portions of these genes or cDNAs to produce hybrid or chimeric forms. This DNA shuffling is done in the hope that some of the hybrid proteins will have unique properties or activities that were not encoded in any of the original sequences. Also, some of the hybrid proteins may combine important attributes of two or more of the original proteins, example, high activity and thermostability. The simplest way to shuffle portion of similar genes is through the use of common restriction enzyme sites, digestion of two or more of the DNAs that encode the native forms of similar proteins, with one or more restriction enzymes that cut the DNA is in the same place, followed by ligation of the mixture of DNA fragments, can potentially generate a large number of hybrids. For example, two DNAs, each of which has three unique restriction enzyme sites, can be recombined, shuffled to produce 14 different hybrids in addition to the original DNA. Excuse me. Another way to shuffle DNA involves combining several members of a gene family, fragmenting the mixed DNA with the deoxyribonuclease 1, that's DNase I, selecting smaller DNA fragments and PCR amplifying these fragments. During PCR, gene fragments from different members or a gene fragment or a gene family cross-prime each other after DNA fragments bind to one another in regions of high homology or complementarity. The final full-length products are obtained by including terminal primers in the PCR. After 20 to 30 PCR cycles, a panel of hybrid full-length DNAs will be established. The hybrid DNAs are then used to create a library that can be screened for the desired activity, a task that may be the most difficult and labor-intensive part of the entire process. Although DNA shuffling works well with the gene families, it is sometimes called molecular breeding, or with genes from different families that nevertheless have a high degree of homology. The technique is not especially useful when proteins have little or no homology, thus the DNAs must be very similar to one or another or the PCR will not proceed. To remedy the situation and combine these genes of dissimilar proteins, several variations of the DNA shuffling protocol have been described. One procedure that was developed to combine the genes of dissimilar proteins and that does not rely on PCR amplification of DNA fragments is called non-homologous random recombination. In this procedure, figure 8.12 DNAs from different sources, either defined or random DNA sequences or a mixture of both, are combined and partially digested with DNase I. These DNA fragments, which include a wide variety of sizes, are made blunt, entered by a digestion with the enzyme T4DNA polymerase. This enzyme both fills in 5' overhanging nucleotides and degrades 3' overhanging nucleotides. The DNA fragment are then mixed with a synthetic DNA fragment that forms a hairpin loop and contains a specific restriction enzyme site before the entire mixture is ligated by the addition of the enzyme T4 DNA ligase to form extended mosaic DNA hairpin molecules are variable lengths. The average length of these hairpin structures is dictated by the ratio between the blunt-ended DNA and the added DNA hairpins, which prevent further concatemerization once they are ligated to the ends. Finally, restriction enzyme digestion removes the hairpin loops so that the resulting sticky-ended DNA fragments can be inserted into plasmid vectors and tested for various activities. Because this process randomly recombines DNA fragments, only a very small fraction of the recombined DNAs are likely to encode the desired activity. Mutant proteins with unusual amino acids. Essentially, any protein can be altered by substituting one amino acid for another using directed mutagenesis. However, this approach is limited to the 20 amino acids that are normally used in protein synthesis. One way to increase the diversity of the proteins formed after mutagenesis is to introduce synthetic amino acids with unique side chains at specific sites. To do this, E. coli was engineered to produce both a novel transfer RNA, tRNA, that is not recognized by any of the existing E. coli amino-acyl tRNA synthetase, or the tRNA synthetases, but nevertheless functions in translation, and a new amino acid tRNA and a new amino acid tRNA synthetase that amino acylates only that novel tRNA. A novel tRNA and unique amino acid tRNA synthetase bear from the Archaebacterium Methanococcus gemnisci was used as a starting point for the system. The tyrosine tRNA synthetase from M. genesi can add an amino acid to an AMBER suppressor tRNA that is a mutant of its tyrosine tRNA. An AMBER suppressor tRNA is a modified tRNA that can insert an amino acid into a protein in places where the mRNA contains an AMBER codon. UAG, which normally codes for a stop, that is the cessation of protein synthesis to prevent translation fusion of of the proteins whose mRNA normally code for a stop. With a UAG with downstream protein, suppression is always less than 100% and is often dependent upon the nucleotides surrounding the stop codon. The amino acid specificity of the tyrosine tRNA synthetase from M. gemnisci is altered by random mutagenesis of its gene, so that instead of tyrosine, it places O-methyl T-tyrosine O-methyl, let me just get that right, L-tyrosine, please, correct me there, onto the tRNA. Specifically, a clone version of the target gene is modified by oligonucleotide directed mutagenesis so that it contains a 5' tag 3' as a portion of the DNA that encodes the amino acid that is targeted for change to O-methyl L-tyrosine. Once the modified DNA has been selected, it is used to transform an E. coli strain that was previously engineered to produce the O-methyl T-tyrosine L-tyrosine tRNA. The engineered E. coli strain inserts O-methyl L-tyrosine into proteins that contain the UAG stop codon, resulting in a full-length target. And the protein containing the modified amino acid, had the mutant gene been expressed in wild-type E. coli, a truncated version of the protein would have been produced. This system may be manipulated to insert a variety of different amino acid analogs into specified sites within proteins in the hope of producing functional proteins with altered activities compared with the native form. In a similar approach to this problem, researchers modified a portion of the valine tRNA synthetase gene so that the altered enzyme was able to insert the non-standard amino acid aminobutyrate into proteins. While the full potential of these approaches has yet to be realized, it is nevertheless clear that it is now possible to produce proteins containing unusual chemical structures and possibly having unique properties. On to the next topic of protein engineering, and before that we have a small figure here which is 8.116 mentions of some of the hybrid DNAs that can be generated during PC amplification of three members of a gene family. So on to protein engineering. But 20 of the many thousands of enzymes that have been studied and characterized biochemically account for over 90% of the total amount of enzymes that is currently being used industrially. Table 8.1 lists some of the most important enzymes that are the primary uses. A major reason why additional enzymes are not used to any generally great extent in industrial processes is that an activity that has evolved to perform a particular function for a microorganism, animal or plant under natural conditions, usually is not well suited for a highly specialized industrial application. Most enzymes are easily denatured by exposure to the conditions such as high temperature and the presence of organic solvents that are used in many industrial processes. Although thermotolerant enzymes can be isolated from thermophilic microorganisms, these organisms often lack the particular enzyme that is required for use in industrial processes. However, the availability of directed mutagenesis and gene cloning, these constraints are no longer significant. Adding disulfide bonds. The thermostability of a protein can be increased by creating a molecule that would not readily unfold at elevated temperatures. In addition, these thermostable enzymes are often resistant to denaturation by organic solvents and to non-physiological conditions such as extremes of pH. The addition of disulfide bonds through the introduction of specifically placed cysteine at residues can usually significantly increase the stability of a protein. The problem is whether extra disulfide bonds perturb the normal functioning of a protein. T4 lysozyme. The 136 variants of the enzyme T4 lysozyme were constructed by oligonucleotide-directed mutagenesis, which introduced the new internal disulfide bonds. Specifically 2, 4, or 6 amino acid residues at a time were changed to cysteine, thereby generating 1, 2, or 3 disulfide bonds, respectively. The side chains of the amino acid residues that were targeted to become cysteine residues were known to be spatially close to each other. The active enzyme, this proximity ensured that the overall conformation of the molecule will remain essentially unaffected by the formation of the new disulfide linkages. The targeted amino acids were not involved in the active site of the enzyme. Figure 8.12 is a schematic representation of homologous random recombination. Different DNAs shown in different colors are mixed together, partially digested with DNase I, blunt entered by digestion with T4 DNA polymerase, size fractionated, ligated with synthetic hairpin DNase to form extended hairpins. Restriction enzyme digested to remove the hairpin ends and generate sticky ends and then ligated into plasmid vectors. And now is the portion of the enzyme molecules that is probably most sensitive to small changes in conformation. The newly introduced cysteines created disulfide bonds between positions 3 and 97, 9 and 164, 21 and 142 of the enzyme, where the numbers denote the amino acid positions in their polypeptide starting from the N-terminus. After mutagenesis, each mutated gene was identified and expressed in the E. coli. The engineered enzymes were purified and the enzymatic activity and the thermostability of each were determined. The thermostability of a protein is often defined as the temperature at which the overall structure of the protein is 50% denatured. The state of denaturation can be assessed by monitoring the circular dichroism of the protein in solution. The wild-type in native form of the enzyme T4 lysozyme has 2 free cysteine residues that both exist as free sulfhydryl groups, neither of which is involved in a disulfide bond. In the so-called pseudo-wild-type enzyme, these cysteines were changed by oligonucleotide-directed mutagenesis to threonine and alanine without altering either the activity or the thermostability of the enzyme. Consequently, the pseudo-wild-type sequence provided a standard for comparing variants with potential thermostabilizing disulfide bonds and also prevented spurious disulfide bonding between the introduced cysteine residues and the naturally occurring ones. The constructed lysozymes derivatives had from 1 to 3 disulfide bonds. The results of this experiment indicate that the thermal stability of the enzyme increases as a result of the presence of disulfide bonds, with the most thermostable variant being the one with the largest number of disulfide bonds, and that some variants C, E, and F in table 8.2, which are more thermostable than the wild type or pseudo-wild type, have lost their enzymatic activity. The loss of enzymatic activity in 3 of the variants probably reflects a distortion of the peptide backbone of the molecule containing a disulfide linkage between residues 2,1 and 142. Often the engineering of a protein is a trial and error process, hence the precise amino acid changes that yield the best variant are not always obvious. However, from this experiment, it is clear that increasing disulfide bonds to enhance protein stability is feasible. Xylanase. So next, in a similar study, the development of a temperature-stable mutant of the enzyme xylanase from Bacillus circularans was undertaken. During the making of paper, wood pulp is chemically treated to remove the hemicellulose that would otherwise contribute to the discoloration of the paper product. Unfortunately, this step results in the creation of large amounts of potentially toxic effluent. From an environmental perspective, treatment of wood pulp with xylanase, which degrades hemicellulose, is preferred to pulping. Treatment of wood pulp with this enzyme could lower the amount of bleaching chemicals that would otherwise be required as a part of this process. However, the step at which xylanase would be added, we'll come to this table later. Its properties are T4 lysozyme and 6 engineered variants. Just after we finish the subject matter proper, follows the hot alkali treatment of the pulp. While it is possible to lower the pH of this material by adding acid, current industry practice is directed toward using less water to cool the pulp. So if xylanase were to be used in this process, it must function efficiently at relatively high temperatures. Computer modeling of the 3-dimensional structure of xylanase was used to predict sites along the polypeptide chain where 1, 2, or 3 disulfide bridges could be introduced in order to stabilize the enzyme without disrupting its catalytic activity. All of the 8 derivatives of the B-cyclins xylanase that were generated showed an increase in thermostability compared with the wild type, and 3 of the 8 mutants were as enzymatically active as the wild type at 60 degrees centigrade. Moreover, one mutant in which a disulfide bridge between the N and C terminal ends of the enzyme was introduced was nearly twice as active as the wild type at 60 degrees centigrade, where the wild type lost all its activity after only 30 minutes at this temperature. These results indicate that the thermostability of other enzymes can be enhanced provided that sufficiently detailed X-ray crystallographic information is available. The success of the protein engineering and the laboratory testing notwithstanding, it remains to be determined whether a thermostable xylanase can be effectively incorporated into a commercial process for the manufactured paper. Remains to be determined whether a thermostable xylanase can be effectively incorporated into a commercial process for the manufacture of paper. Next is human pancreatic RNase. Ribonuclease RNase from bull semen can act as an anti-tumorigenic agent. In both in vitro and in vivo experiments, the dimeric form of this protein is internalized into tumor cells by a non-receptor mediated endocytosis, and when it reaches the cytosol, it selectively degrades the ribosomal RNA, that's rRNA, thereby blocking protein synthesis and causing cell death. The dimeric form of this enzyme consists of 2 identical subunits covalently joined by two adjacent inter-subunits disulfide bridges and stabilized by a small number of non-covalent interactions. The antitumor activity of BUL7 RNase is dependent on the dimeric structure of the protein. Figure 8.15 shows the addition of a disulfide bond between N and C terminal of B circulins xylanase stabilized. This is the protein. Its activity at room temperature is doubled, and at 60 degrees centigrade it is largely protected against heat inactivation. Activation room temperature, look at the figure and take a screenshot. Make notes while you prepare. The oligomeric RNase from the pancreatic-like RNase superfamily of proteins. However, human antibodies against BUL7 RNase are often produced following repeated or prolonged use of this therapeutic protein, thereby eventually negating the usefulness and effectiveness of BUL7 RNase in treating tumor cells. For this reason, monomeric human pancreatic RNase was engineered to be an anti-tumorigenic agent. The amino acid sequence of BUL7 RNase is more than 70% identical to the amino acid sequence of human pancreatic RNase, and therefore could be used as a model to determine which amino acids might be altered in the human enzyme to participate in dimer formation. When dimeric human pancreatic RNase was found in E. coli, the protein was localized in the insoluble inclusion body. Solubilization of the inclusion body and renaturation of the unfolded protein yielded dimeric human pancreatic RNase that displayed a slightly lower level of anti-tumorigenic activity than BUL7 RNase. So depending on the tumor cell line, up to twice as much of the engineered protein was required. Since the dimeric human pancreatic RNase did not impair the functioning of normal human diploid fibroblast cells, this engineered protein is a good candidate to become an important human therapeutic agent. Which one? The dimeric human pancreatic RNase is considered a good human therapeutic agent as it does not impair the function of normal human diploid fibroblasts. On to the next subheading, changing asparagine to other amino acids. Change in asparagine is like when proteins are exposed to high temperatures, asparagine and glutamine residues may undergo deamidation, a reaction that releases ammonia. With the loss of the amide moiety, the amino acids become aspartic acid and glutamic acid respectively, which have different chemical properties at physiological pH than asparagine and glutamine, resulting in localized changes in the folding of the peptide chain that may lead to a loss of activity. In one study, the enzyme of effective changing some asparagine residues in the Saccharomyces cerevisiae enzyme triose phosphate isomerase was examined. This enzyme consists of two identical subunits. Each subunit has two asparagine residues that will contribute to the thermosensitivity of the enzyme because they are located at the subunit interface. But oligonucleotide-directed mutagenesis, the asparagine residues at positions 14 and 78 were targeted for change. Converting either of these asparagine residues to threonine or isoleucine enhanced the thermostability of the enzyme, whereas predicted. Changing one of the asparagine residues to aspartic acid reduced thermostability. When both asparagine residues were changed to aspartic acid residues, the resulting enzyme was enabled even at ambient temperature and had low enzymatic activity. The engineered proteins were also tested for their sensitiveness to proteolytic digestion. A positive correlation between thermostability and resistance to proteolysis was observed. On the basis of these results, it should be possible to generate thermostable forms of other enzymes by mutating non-essential asparagine codons. In fact, a long-acting human insulin analog was produced by replacing an aspartic acid residue with glycine. This analog was recently approved for use as a human therapeutic agent. The next topic is about reducing the number of free sulfhydryl residues. Occasionally, an expressed foreign protein is less active than expected. Protein engineering can be used for increasing this activity. For example, when the cDNA for human IFN beta, interferon beta, was initially cloned and expressed in E. coli, the protein product showed only a disappointing 10% of the antiviral activity of the authentic glycosylated form, and although reasonable amounts of interferon beta were synthesized, most of the IFN beta was found to exist as dimers and high oligomers that were inactive. Analysis of the DNA sequence of the IFN beta gene revealed that the native protein has 3 cysteine residues, so one or more of these genes could be involved in the intermolecular disulfide bonding that produced the dimers and oligomers in E. coli, but not in human cells. It was reasoned that conversion of one or more of the cysteine codons into serine codons might result in an IFN beta derivative that would not form oligomers. Serine was chosen to replace cysteine because the structures of the 2 amino acids are identical, except that serine contains oxygen instead of sulfur, and as a result cannot form disulfide bonds. When the study was undertaken, detailed information about the structure of IFN beta was lacking, so the researchers were forced to rely on data derived from related proteins. In other words, they did not know which of the 3 cysteine residues contributed to intermolecular disulfide bond formation. However, the locations of the cysteine residues that form the internalized disulfide bonds IF and alpha, a structurally similar molecule, were known, so as a comparison of the primary amino acid sequence of this molecule. These tables will be referred to later. The molecule with that of IF and beta was made as seen in figure 8.18. Alignment of the amino acid sequences of the 2 protein molecules indicated that cis31 and cis141 in IF and beta are located in positions similar to those of cis29 and cis138 in IF and alpha. Because of cis29 and cis138 in IF and alpha are involved in the formation of an intramolecular disulfide bond, it seemed reasonable to assume that cis17 of IF and beta was not involved in the intramolecular disulfide bonding and was therefore a good candidate for modification. This deduction proved to be correct. No multimeric complexes were formed when a serine 17 variant of IF and beta's serine 17 IF and beta was expressed in E. coli. Moreover, the serine 17 IF and beta had a specific activity similar to that of authentic native IF and beta and was more stable during the long-term storage than the native form, increasing enzymatic activity. In addition to stabilizing an enzyme by directed mutagenesis, it may be feasible to modify its catalytic function. Currently, detailed information about the geometry of the active site of a well-known characteristic enzyme required in order to be predictably alter enzymatic activity. With such data, researchers are able to deduce which specific changes might be necessary to modulate the substrate binding specificity of an enzyme. We're on to the next topic, tyrosine tRNA synthetase. Now, an early demonstration of how directed mutagenesis could enhance enzymatic activity. The enzyme tyrosine tRNA synthetic activity from beta stereothermophilus was modified with respect to substrate binding. Tyrosine tRNA synthetase catalyzes the amino acylation of a tRNA that specifically accepts tyrosine tRNA type in a two-step process. 1 tyrosine plus ADP gives tyrosine A plus PP. 2 tyrosine A plus tRNA TYR tryptophan. TYR tRNA gives plus AMP. In step 1, tyrosine, this lecture will be over in a minute and we'll be continuing in the next lecture, is activated by ATP. It yields an enzyme-bound tyrosyl adenylate, tyrosine A with the concomitant, right?